This video is looking at the topic B1, cell biology, focusing on stem cells. The first thing to say about stem cells is that they are undifferentiated. That means they look like our basic animal cell, our basic plant cell. Okay, they haven't become specialised. So think of differentiate as becoming different. So they are basic cells that can become potentially lots of different kinds of cells. In animals, differentiation happens very early on, and in plants, it can happen at any point in time. So we've got three sources of stem cells in humans. First of all, we've got bone marrow, which can sometimes be referred to as adult stem cells, but this doesn't mean that they have to just come from adults. It means that they don't come from a small embryo. Now, bone marrow is a difficult operation. It's quite painful. So your bone marrow is drilled out through your bone, so the bone marrow is inside the bone, drilling into the bone to remove the bone marrow. What's really useful about this, a really good advantage, is that you can make blood cells from it. There's only a few types of cells you can make, and it's painfully extracted and can result in maybe an infection afterwards when it's healing. The second approach is using embryos. Now, we're not allowed by law to make embryos just for stem cells. The only embryos we can use are those that are medical waste from IVF. IVF we learn about in topic B5, and it's basically where the egg and the sperm are fertilised together outside of the body in a petri dish. You might have heard them being called test tube babies, and if there are any embryos left and the couple have had the number of children that they want and they don't want any more of them, then they can decide to either destroy those embryos or donate them to science to be used as stem cells. So the good thing about that is an embryo, a fertilised egg, can be used to make any kind of cell we need. So it's really, really useful in terms of making lots of different kinds of cells. But there are ethical issues where people say that it's destroying a potential human life and that we shouldn't use them from embryos. The third option, which I think is really exciting, is the use of umbilical cords. So our umbilical cords are long gone, but by the time you have children, you could be asked if you want to store the umbilical cord so that your child could use stem cells from the umbilical cord in the future if they need them. The good thing about this, if you use your own umbilical cord, is that your body will not reject any of the cells, but the bad thing is that you'd have the storage issues of the cost of storage and the space for storing them in the future, because if everybody starts storing their umbilical cords, it would take up a lot of space. Then we've got the last option. This is where we make what I call an ethical embryo. This is called therapeutic cloning of an embryo. So we learnt that embryos can make any kind of cell, but we know that that's unethical because it's a potential human life. But what if we could make an ethical embryo? We'd get any kind of cell, but it wouldn't have those ethical issues. So it's a bit like cloning, which we learn about in B6. So let's say the patient needs some skin cells. Maybe they've got some burns. Here are some of the skin cells taken from that person. You then take an egg cell. Why? Because an egg cell can become an embryo. You remove the nucleus because you don't want the genetic information from the egg in that cell. All of the genetic information is available in pairs in these skin cells. So we take the nucleus out of the egg and then we fuse the two together. Okay, so here we've got these skin cells, the nucleus from them, in this empty egg which came from over here. Then that's stimulated, usually using an electric shock, and it starts to go through mitosis, also covered in B1. And what happens there is you just multiply the cells, and then you can get lots of cells that are all the same, that can then literally be used like a spray paint, sprayed onto the area that needs new skin cells. And they've come from an embryo, but it's an ethical embryo. So that's called therapeutic cloning of an embryo. Let's look at those stages again because it's quite a difficult process. So the nucleus is taken out of the human egg cell. The cells that you would like to recreate are put into that empty egg cell. It's stimulated to develop into an embryo, so it goes through mitosis, so it's started to multiply, to go through cell division. And then stem cells are taken from that embryo, and I didn't say in the first bit, but I'll say it now. Those stem cells are then grown, so they continue to multiply in a container of warm nutrients. Those stem cells can be used to treat whatever they need to be used for. The last one to say then is that plants have stem cells too, and stem cells in plants are called meristems. And we said earlier in the video that stem cells in plants can differentiate at any point in their life. So you could have an old oak tree that's hundreds of years old and it can still differentiate cells using those meristems. Why would we want to use stem cells from plants? Well, we might want to clone some rare species of plants so we can take the stem cells and use them to grow more of them. It might be that we have a plant that can give us a particular medicine and we need lots of that medicine, so that we could use meristem cells to get lots of that particular plant for the medicine. Or it might be that they have some other kind of special feature that means we would like to have lots of them. Hope you found that useful. Thank you.